again, thank you everybody who's online for signing up and attending the IWGSC webinar series. We'll very soon be joined by Dr. Ping Yang for a very interesting talk. Before we start, I wanted to give a few words of introduction about the IWGSC. We are an international nonprofit organization with a goal to increase wheat science. We have 3,500 general memberships. We are present in 80 countries and involving 985 institutes or companies. And we have 6 sponsors that I want to mention that help to fund webinars such as this. And they include Arbor Bioscience, CIMMYT, INRA-E, RAGT, Kansas Wheat, and Florimond Desprez. So thank you very much, sponsors. So the IWGSC was responsible for the first reference genome released in 2017 and published in 2018. Since that time, we've updated our vision, our 2.0 vision, to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And in order to accomplish this goal, we are now engaged in the Wheat Genome Variation Project, where we are doing complete sequences of 12 very highly diverse wheat landrace successions. And we are also improving the Chinese spring reference sequence. So this project will be releasing transcriptome data, gene annotations, updates, and also all of the data will be made available in a structure, called a practical haplotype graph, which should be very good for performing genotyping. So I just want to acknowledge the National Science Foundation and the European Union for funding and our collaborators shown here. Upcoming IWGSC events. So there'll be another webinar coming up on in December 11th, and will be met by Vijay Tiwari, speaking about ancient grain genomics to simplify gene discovery and deployment in wheat. In addition, so please sign up and join for that. In addition, if you are attending PAG in San Diego in January, we will be hosting the IWGSC workshop on Saturday morning. Okay, and now to some business about this webinar. So just so you know, the webinar will be recorded and posted on the IWGSC YouTube channel. And as Ping Yang does his talk, if you have questions regarding the talk, please press on this Q&A button, and you type in your question at the end of the talk. Given enough time, I will read the questions for Ping Yang to answer. There's also a chat button. This is mostly used if you have some kind of technical difficulty, or if there are handouts or other information that are being provided to you. Okay. And now let's get to the main event. So Dr. Ping Yang from the State Key Laboratory of Crop Gene Resources and Breeding, Institute of Crop Sciences, and the Chinese Academy of Agricultural Sciences. He will be speaking on the exploration and precision editing of host factors to develop Bymovirus resistant cereal crops. So thank you very much.
Yeah. Can you see my screen?
Yes.
Okay. Okay. First of all, I would like, should I start now?
Yes.
Okay, okay. Yeah, so before I start, I would like to firstly thanks the International Weight Genome Sequencing Consortium for the invitation, and also thanks the Josh for the kind introductions. Today, I would like to give some to introduce to some work in my group working on the virus resistance. Um, so we against the barley Bymoviruses in barley and wheat. So my name is Ping Yang from the Institute of Crop Science, Chinese Academy of Agricultural Science. So as you can see here, so this picture is a diseased field. So in Germany, which is kindly provided by the Dr. Frank Robinstein from the GKI in Quedlinburg. So this is a barley infected field by the barley yellow mosaic virus. As you can see here, so the yellow part basically is the virus infected um, field. So before I start, I would like to briefly introduce where I'm from. Now I'm working in Beijing. Basically, you can see here map is here. So and this second picture is our working buildings. So this is a National Crop Gene Bank of China. So this is a new building. Is established 3 years ago. So this is a view inside. Basically, this drawer is the weight architecture, and here there is a middle of the ground. So there is one word which is in historic Chinese means 种, in English means seed. So that means our major goal is to collection and maintain the germ presence for the future to ensure the future food securities. So this is my group. Basically, is so my group is barley genetic resources, and our mission is to characterize and to make innovation of the barley germplasm resources. Because barley and wheat are closely related species, so once we found some interesting work, we are also extend our observation to wheat research. So today I would like to introduce three parts. So first to introduce the soil-borne Bymovirus diseases, mostly in barley and few in wheat. And secondly, based on our observation in barley, we want to extend to our work in bread wheat. And finally, would like to provide a perspective how to exploit the lower susceptibility factor genes for the future to enhance the disease resistance. As you may hear, the in cereal crops, so the fungi and bacterial diseases are often mentioned. However, the virus diseases are also one of the major threats in the cereal crops. As you can see here, for instance, 70 years ago, the barley yellow dwarf virus occurred worldwide, and last in the 1970s to 1990s, the barley yellow mosaic virus occurred widely in East Asia and also most of European countries. So I just briefly collected the epidemic of the virus disease in the cereal crops in the last few decades. So if you consider the virus with compared with the bacteria or the fungi pathogens, the major difference, the virus is obligated. So they are not survival independent of the host species. So that's why, so the virus, if would like to successfully infect the host plants, there so the vectors would be is required. So more than 80% of virus are basically transmitted by the insects. In another case, So around 80% of the virus are transmitted by the fungus. So today I would like to introduce our work, mostly our work on the fungi Polymyxa graminis transmitted fungus. So as you can see here, this is the panel. So the barley yellow mosaic virus, this basically is the typical virus strains of the genus Bymovirus. So this virus contained two RNA strains. So in totally encoded 10 or 11 virus proteins. So that means the virus have a very small genomes, therefore encoded very few number of proteins. And also the virus is obligate, as I mentioned, they are not able to survive without the hosts. In another hand, maybe also the serious case. So virus once it's infected or established infections, so the chemical treatment becomes useless. So that means virus protection is mostly related on the host itself instead of the chemical treatment. So for the soil-borne, virus is transmitted by Polymyxa graminis. Basically, as far as we know, there are included two genus. So one from the Petroviridae, the family of the Petroviridae, the BaMMV. Another is fluvirus. So there are more than 10 different streams. So this virus are able to infected so wheat, barley, oat, triticale, rice, and also sorghum. Specifically for my interest, I'm working on the Bymovirus. So this Bymovirus, so there are four different streams: barley yellow mosaic virus and barley mild mosaic virus. So both viruses are specifically infected barley. Another two viruses is wheat spider streak mosaic virus and also wheat yellow mosaic virus, both specifically infected wheat. So, uh, so um, so this is basically the disease field. So this one I already show you in the first slides. So this is another pictures from the central China. So this area is basically the main wheat producing area in China. So this is the field infected by the wheat yellow mosaic virus. So the wheat yellow mosaic virus is occurring every year in China and with the largest rate. So if you only consider the virus infections in wheat. And for the barley, so this barley yellow and barley mild, both are serious threats. So for more than 20 countries in Europe and in East Asia. So this is the picture. So basically mention the countries where the virus were reported. As you can see, so the barley yellow mosaic virus and the barley mild mosaic virus are mostly reported in European country and in East Asia. So a few years ago, this barley yellow mosaic virus strains was also reported in Iran. So for the wheat yellow mosaic virus, this disease is often reported in the East Asia, China and Japan, and also in the India. For the wheat WSSMV, so this is only reported in the literature, so in several European or North American countries, and also one case in South Africa. So this is the epidermal of the viruses in China. So as you can see, the map, basically the green indicated province are wheat producer, main wheat producer in China. So the red dots indicate this site were reported with the wheat yellow mosaic virus. So especially at the south part of Henan province. So this area are so this wheat yellow mosaic virus is a major threat for these areas. And also you can see another virus, so the Chinese white yellow mosaic virus. Basically, it's not belong to the Bymovirus, it belongs to the fluvirus, are reported occasionally in the different site in China. So the barley yellow mosaic virus and the barley mild mosaic virus, it was a big problem in China if you consider the 3 or 4 decades ago at that moment. So the barley is considered one of the major crops around the Yangtze rivers. But however, in the last 20 years, the barley growing field in China decreased constantly. So therefore, in this area, so this virus is only observed in the at the downstream of the Yangtze River, specifically in Jiangsu province, close to the coast. So based on our experience in our group, we are working on the 3 streams. So barley mild mosaic virus, yellow mosaic virus, and also white yellow mosaic virus. So both these 3 viruses are all transmitted by the soil-borne fungi polymyxogrammes, which is basically underground. So after the sowing in waters, so the fungal spore germinate is able to infect the seedling roots and transmitted the virus components into the host species. So during over the winter, so the virus particles move slowly to the younger leaves in the early springs. That's why usually in the early spring in February or the early March, we are able to observe the virus symptoms in the youngest leaves. So this is a relationship between the virus and the vector and the host. So the virus is present in the fungus, and the fungus is able to infect the host and help or transmit the virus particles into the host. So in another hand, for the research, we can also use the mechanical inoculation. So for using virus swab to inoculate the host plants, therefore to test for resistance. So for the plant protection, so basically, so improve the resistance in the host species is feasible. So that means resistant breeding is the optimal strategy to prevent the virus threat. So this is symptoms in the leaves. So this is the barley mild mosaic virus infected leaves, and this is healthy plants. So you can clearly see, so the infected plants show the yellow discolorations and also the necrotic pitches at the late stage. In order to identify the resistance, so the barley geneticists and also pathologists spend many years to identify the host resistance against the virus strains. So 20 years ago, so there is 2 groups clone the first susceptibility factor genes in barley. So this encodes eukaryotic initiation and translation factor 4E. So this protein is required for the virus protein translation initiations. So there are 2 isolates from the host species, RUN4 and RUN5. Both are resistant allele. If you look at so this compared to the susceptible lines, the resistant locals have several amino acid substitutions at that moment. So my former boss, so PhD supervisor Neil Stan, and also my former collaborator Kostia Kanyuka, they use a map-based cloning and the candidate approach to clone these genes. So 20 years ago. So when I started my PhD in 2010, so with Nielstein, so we are considered to clone another recessive genes, RAM11s, because the RAM4 and RAM5, both locals were widely used for barley breeding. However, several resistant breaking isolates appeared. So in Europe and also in Eastern Asia countries, therefore identified lower resistant resources becomes of interest at that moment. So use the bar-based cloning approach, we finally clone these genes. So these locals RAM1 and RAM11 basically are allelic, and these locals if so in the resistant lines, we observe large deletions in the promoter and the first three exons. So this deletion leads to the complete lost function of these genes. So therefore, no translation at all in the resistant lines. So so if you consider both genes, so EF4E, the resistance are from the amino acid substitutions, and for the PDF5-1, the resistant variants were from the loss of function of the susceptible genes. So therefore, we believe at that moment, so EF4E becomes useful because it's considered important for the protein translation initiation. So we think this maybe is EF4E is dispensable, but this PDF5-1 is dispensable. So, several years later, so we are considering because the genome editing becomes popular and not only in the model species and also becomes popular in the crop species like wheat and barley. So my former collaborators, Johan Kuhlmann's group, so they conducted the genome editing in the host factor genes EF4E. Very surprisingly, they are able to obtain the say loss functional allele. So that means this host factor even is important for the protein translation, but is dispensable. So and the lockout of the EF4E genes leads to the resistance to the mild mosaic virus. So E-gray is the wild type, and you can see here three independent edited lines show the complete resistance to mild mosaic virus. Almost at the same time, we are working. When I start my job in 2017 in Beijing, we trying to edit the another gene PDF-I5-1 in barleys. So as you can see here, we also have obtained several different edited lines compared to the susceptible lines after inoculation. The edited lines so show the complete resistance for virus inoculation. So basically, this result indicates the both genes are reliable, and also we are able consider we are with the newly developed biotechnologies, we are able to generate resistant lines in the by the genome editings. So in another hand, we are also consider so the wheat because in China, wheat is more and more important than barley. So and also wheat yellow mosaic virus is a major threat. So in China as well. So we are considered. So at by now there are three resistant genes reported in wheat. So three of them are encoded by the dominant genes. For instance, this gene cloned by the Takao Komatsuda YM2, so this is NBSLR genes, and another NBSLR genes encoded for the YM1, and also another small peptide is considered also close to the this major locus, so on 2DLs. So as you can see here, this resistance in wheat are mostly come from the wild relatives. So so this is somehow is different to barley. So now I would like to switch to my second part. So as I mentioned, if you consider wheat and barley, so wheat is a hexaploid, barley is diploid. If you consider search the literatures, so to identify the resistance locals against the wheat yellow mosaic virus or barley yellow mosaic virus, we can see a very different. So in the wheat, there are 7 genes at the 7 locals were reported, but none of them are inherited by the recessive genes. So all the genes are dominant. But in barley, it's different. Out of 22 genes, 19 genes are recessive. So if you consider the barley and wheat, one is hexaploid and another one is diploid, we simply come to this hypothesis. So because we speculate the polypolarization in wheat has resulted in functional redundant genes, therefore block the identification of recessive genes. These recessive genes are derived from loss of the susceptibility genes. So, for instance, in barley, if one gene is loss of function, we can easily maybe this either to exploit the genetic effects, but in the wheat, so it would be becomes very rare to simultaneously loss of three genes. With this hypothesis, we use genome editing to look out the PDI5-1 genes in wheat. At that moment, we get four independent edited lines. So that means the three copies from the A, B, and D genomes are simultaneously looked out in each single plant. So we use a virus inoculation. So the edited lines show the complete resistance. So that means we are not. So the virus particles is lower than the detection limited. We also use the qPCR to quantify the virus particles. As you can see here, the susceptible line and the resistant lines, so they are more than 10,000 fold different. So that means if we look out the PDF5-1s, so from the A, B, D simultaneously, we are able to produce complete resistance lines against the virus infections. So also we conduct the crossing because we want to check the genetic effects. If we only look at the genes in A genome, B genome, or D genomes, so therefore we finally get the several single mutant or double mutant. So look out the one or two copies from the three subgenomes. So as you can see here, also, so the lines after inoculation only the triple edited PDF5-1 produced resistance, but so the single mutant or double mutant, so we are able to look out to reduce the virus particles if we compared with the susceptible lines, but still these plants remain susceptible. We are able to observe the disease symptoms in the younger leaves. We also conduct the same work, so switch to the another host factor genes EF4E. Basically, we follow the same strategies, and also we observed only if we all simultaneously look out three copy EF4E genes, we produce resistant lines. Otherwise, the single or double mutant remains susceptible. We also analyze the agronomic performance at several traits. So this is basically the pictures from the single or double or triple mutant from the EF4E locus. So this one is a triple mutant. So if you compare the triple mutant and the wild type, so basically, we didn't observe the yield penalties, but however, the triple edited plants is tall and also becomes late maturities. So this basically is considered the disadvantages pleiotropic traits. So for the breeding use. Also, we did the same things for another for the PDF5-1. So as you can see, very surprisingly, and also not really surprising, but is under expecting, because based on our experience in barley, so lots of triple edited PDF5-1 in weight. So the edited lines basically show the similar performance compared to the wild type. So that means this locus PDF5-1 is considered a good target for genome editing for white yellow mosaic virus resistance. However, so the genome editing is considered, especially in China, is considered is transformation associated technologies. So this is still under serious biosecurity controls. So therefore, the genome edited lines are not directly released to the field, and therefore we are considered the alternatives. So as early to the 2021, we conducted mutagenesis in one with major cultivars. So we spent 1 year for the mutagenesis plus the half year for the specific target identification the mutation at the PDF5-1 locus. And plus then we made to cross the each single mutant with 2 generations with marker assisted stacking. So we get the at the beginning of last year, so we get the triple edited lines at the PDF of the locus. And after 2 back crossings in half years, and also 1 generation selfings, we finally get the triple edited line, not triple edited line, triple mutagenesis lines. So in this weight of varieties. So, but still we are also trying to analyze the agronomic performance. So the advantage of using the mutagenesis lines, we are not so because the mutagenesis is considered a normal breeding approach. So we can futurely we can release these lines for in the wheat breedings. As a summary for this part, so I would like to highlight what we have done in the barley and wheat, especially to targeting the PDF5-1 locus for the resistance against 3 virus streams. So if we, for instance, in the future, if we want to generate resistant lines, we can use the genome editings. So based on our experience, we can generate the edited lines without the plasmid fragments, without the off-tags, and also no penalties observed. And once the explants available, we can get the improved lines in 1 year if we use genome editings, both in barley and in wheat. On the other hand, we were also able to do the marker-assisted selections. So, however, so because especially for barley, it's okay because we can only transfer integration one locus. It's easier, and also, but for the wheat, so because we have to simultaneously integrate 3 fragments from 3 genomes, so it will take many generations back crossing and selections. However, we are also not able to, if we use marker-assisted selection, we are not able to exclude so the linkage drug because this, but however, this is genotype dependent. Genotype dependent. So once, for instance, with our technologies, I would definitely prefer to use genome editing to generate resistant gene plugins for the futures. That would be much easier. So now I would like to switch to my last part. So as you know, so the host factor gene, as I indicate, is very important. Is very useful to generate resistant lines, especially you combined with the genome editing technologies. So the major interest is how to identify the novel host factor genes. So if we look at these genes, we are not bring disadvantages performance on the agronomic trees. So therefore we are considering how to use our experience in barley and wheat to identify the novel susceptibility factor genes. So before I start my job in Beijing, I considered to generate the mutagenesis population in barley. So this way, at that moment, we selected one line which is Hati Xi. So this is the Chinese land races. this is a very special isolates because once this plants get maturities, so you can see the spikes, arms, and also the leaves becomes complete black. So and also this lines is susceptible to virus inoculation. So that means so if we are able to identify the loss of susceptibility in this mutagenesis population. Sorry. So therefore, using EMS assisted mutagenesis and to generate M1 generation, and we use a single spike um, descent pipelines to generate the M6 generation plants. So would like to grow in the M because at the M6 generations, most of locals becomes homologous. So therefore, we are able to use this line to test for resistance in the field. So and also we have the each spike from the one of the one plants in M6 generation for the M7. So this line will have several duplicates to for the field trail again. So therefore we are able to identify the resistant plants with these technologies. So our target is we want to I'm sorry. We want to identify the resistant lines out of the susceptible mutagenesis populations. So firstly, we have to exclude the variants which be caused by the mutations at both susceptible locals. Therefore, to identify the novel variants. Once the lines are identified, we will use the whole genome shotgun sequencing and plus the mutamap strategies to identify the new S genes. So once we confirm this observation in barley, we will switch to wheat. Use the genome editings to look out the homologous lines, homologous genes in wheat. So in order to conduct this experiment, we have to find the desirable site for the virus testing. So many years ago, we searched for the virus and also virus distribution in China. So we finally observed one site which only infected by the barley yellow mosquito virus and not the infested by barley mild mosquito virus. So this loci is from the Dazhong National Farms, so in the Jiangsu province. So once we found this place, we since 2022 we growing the susceptible cultivars in this field. As you can see here, so this is the infected lines barley, and the far here is wheat. So wheat is free of the barley yellow mosquito virus infections. So that means we are able to basically to normalize or to optimize the virus distribution in the field. So we grow in the susceptible one for 3 years in the same locals. So therefore, in last year, so we grow in the sowing the mutagenesis population. So in these locals. So this picture was taken last winter in the Dazhong National Farms. And so this is picture we observed in the February this year. So this is basically susceptible lines. This is resistant lines. With our preliminary result, so we out of the more than 8,000 M6 lines, we get more than 200 plants show the resistance. However, because the virus escape of virus inoculation, so in the field is common. So I speculate here there is more than 60% of lines remain susceptible. So that means we harvested this resistant the resistant lines for the field trial this year, And also we harvest each single spike from the more than 8,000 lines with 4 different replicates cultivated in the same site. We want to identify the resistant lines with this experiment. So therefore, once we, as I mentioned, if we get these lines, we will use use the classic strategies to clone this susceptibility factor genes in barley. So this is my last slides. So I hope with my slides I'm able to show you a story about the the Bymovirus resistant genes in barley and wheat. So with our result, we found the PDI5-1 and EF4E are host factor genes for the 3 isolates. So barley yellow mosaic virus for wheat, and barley yellow mosaic virus and barley mild mosaic virus in barleys. So specifically, this result indicate. So in the barley and wheat, basically there is common strategies for Bymovirus infections. So in addition, so with the genome editing result, also with because the PDF5-1 resistant variants RAM1 or RAM11 are commonly used in the barley breedings in the East Asia. So for the more than 2 to 3 decades after use, we know loss of these genes in barley is not bring the disadvantages changes in the performance. So therefore, we believe the PDF5-1 this is a desirable target for enhance the resistance in barley and wheat. So we can and without the yield penalties. So we can use genome editing, and also we can use the marker assistant select transload integrations. So in the future, so what we with our mutagenesis preparation, we want to clone more host factor genes to the barley mild mosaic virus, barley yellow mosaic virus, sorry, and we will identify to analyze their functional mechanism. I think this would be not only interesting in barley, but also was interesting in wheat. So finally, I would like to thanks my colleagues. So what I mentioned here basically is from my 4 colleagues. So the genome editing in barley and wheat were conducted by my colleagues Dr. Jin Hong Kan, and my two former master students Yu Cai, and also Chunyuan Chen. Yu is working with Dr. Doran Petrovich in GKI for his PhD right now, and Chunyuan is working on another institute for his PhDs. For the mutagenesis populations, this was conducted by my colleague Cheng Jiang. So yeah, I would like to appreciate their efforts to work on this excellent on these projects. And also, I want to thank my collaborators, Huiquan Shen, and also Xinguo Ye. Huiquan is help us to conduct the field trial in Yancheng City, and Xinguo Ye and contribute for the genome editing. And Zheng Tianhe is also help us to conduct the field trial for wheat yellow mosaic virus disease. And also, I would like to appreciate my former supervisor, Neil Stein, and also my collaborator, Frank Audens from Germany. Finally, I would like to appreciate my funding agency to support me to work on this virus resistance in barley and wheat. And also, thank you for your attention, for all your audience.
All right, thank you so much. That was a great talk. We have time for question and answers. If you have a question, please go to the Q&A icon and type in your question. If you'd like to vote upvote particular question that in order to prioritize, we could do that as well. So I'm going to read the questions for Ping. So the first question is, I was wondering, has there been any investigation into the genetic diversity of P. graminis population, and how is this diversity might influence virus transmission efficiency or host specificity across different cereal growing regions?
Yeah, so this is a very excellent questions.
And I think this is also related to the second question is might be similar. I'll just read that quickly. It says, since polymyxograminous also transmit oat mosaic virus and oat golden stripe mosaic virus in oats, I was wondering if there have been any studies comparing the interaction of transmission mechanism of these oat infecting viruses with those of the viruses that you just discussed. Okay. And also, could differences in P. graminis genetic diversity influence host virus relationships? Yeah, so those two questions are kind of related.
Yeah. So for the transmission fact the vectors polymyxograminase. So as far as I know, so there is one Chinese group. So they Jian Pingcheng. So he was also working in the virus resistance, performed very well work in this in the passing side. So they investigate the fungi populations. So by collected samples across the different locations in China. Uh, as far as I see, so there is different types. But I because you know some fungi pathogens are not carrying the virus streams. They are not able to test it. So if several specific pathotype would be with the virus would be more has higher infection efficiency or not. As far as I see, I didn't see such reports. And also we last before last year we conducted another experiment to use the different virus to infect the to use the three virus white yellow mosquito virus, barley yellow and barley mad mosquito virus to infect the different hosts. So, so therefore, at that moment, we also not only to check the virus, we also check the polymerase granules. So as far as I know, so and also based on the former report, so the polymerase granules is able to infect hundreds of the treaty species. No, as far as I see, there is no exceptions. So that, however, so this polymyxogram is not is not considered the pathogens for the host species because we do not observe the difference. So the polymyxogram infected or not infected, the host plants show the different performance. Yeah, that's what.
Okay. All right. Thank you. Uh, Second question, are you aware of any resistance genes for wheat spindle streak mosaic virus in wheat?
Yes. So, so there in 2020, I was invited to submitted review articles in TAG. So we summarize the resistant locals in the against the different Bymovirus. You can check the review articles. As far as I say, there is one famous gene, WSS1. So this is resistance to the this virus. And also I want to mention, so if you consider the WSSMV and white yellow mosaic virus, barley yellow mosaic and mild mosaic virus are from the same genus. So I speculate that these viruses have the same mechanism to infect the host species. So that means with this speculation, it's worth to test it, the PDI and E4E triple acid lines with the WSMV But however, in China, we are not able to conduct it because this virus is not available in China. And so many years ago, Costia contacted me. So we consider to do the infections, use use both viruses, WSMV and also WYMV. However, at that moment, finally, we didn't move forward on that collaborations. Yeah.
Okay, thank you. Next question. I'm not clear on this question. It says, were you able to identify any particular transcription factor that worked in both barley and wheat?
You mean the transcription factors, uh, so as a host factor genes, right?
I guess so. Yeah.
Uh, I think yes. So especially I believe there are some transcription factor genes contributes to the virus infection or virus replication or movement, whatever. So, and also if we look out some genes with our unpublished results, we know so the virus um, so this transgenic line shows the difference on virus accumulations. So that means such transcription factor genes are related to the virus infections. As far as I see, we do not have the direct evidence. So that means to look out or to overexpression transcription factors, genes will is able to confer complete resistance as strong as the EF4E or PDF5-1. I didn't observe such examples yet.
Okay. And this question, what approaches have you been used or have been used to confirm that edited lines were free from CRISPR-Cas
Yeah,
contamination?
we conduct the whole genome shotgun sequencing.
Ah, okay.
Yeah.
Okay. So those were the all of the open questions. I have one more question. I'm just curious about the how much you are learning about the molecular mechanisms of susceptibility. And in particular, I'm wondering if it is known whether any of the viral factors, effectors, or proteins interact with molecularly with susceptibility factors, and if so, would it be possible to knock out the susceptibility function without knocking out the wild type host function?
Uh, yes, I think this is a very good speculation, especially consider the EF4E genes, because triple edited EF4E uh, do result in the disadvantages performance. So that means so the EF4E is definitely is important genes.
Right.
For the PDF5-1, this might be exceptions because these genes belong to the small gene families. We know there are 10 different family members. So this is smallest one in this family. And I believe so the different family members have the functional redundant. So that means if you look at one genes, so the other members maybe it take over the their functions in the plant side. So that means we are not able to observe the some disadvantages in the host species.
So there
So
might be some molecular differences between members of the gene family, some of
yeah,
which interact with the virus, others do not.
yeah.
Okay, all right, that's
So
very
this, so
interesting.
that's why this members is very special members. So so only this one is able to interact with the virus. So and also I think this is uh, less important members compared to others. I think this is also smart for virus. So for instance, if the virus trying to hijack important members in host plants, so this is not smart for the virus itself. They will
Yeah.
should differ. Yeah. And as for you mentioned your basically your question regarding to the mechanisms. So if you look at the symptoms, so the virus infected show the discolorations, basically yellow leaves. So basically, so this symptoms is associating with the damage in the chloroplast. Yeah, in the
Yep.
so we with our result we identified one genes. So so from this complex, so is us able to interact with the PDF5-1, and I believe this gene is very important. So, but still we are working on the functional mechanisms right now. Yeah.
Okay. Uh, there's one, maybe if there's enough time, there's one more question just popped up. Okay, are you aware of any whole genome sequences of the polymyxa graminis? Any genome
Yeah.
sequence?
Yeah, yes, definitely it's available. So found on the website, you can find it. So because the polymyxa graminis have the small genomes, so because if you compare to the host species, so you can sequence the infected roots and to so you can sequence the pathogen, not the pathogen, so the vector plus the host species together. So you can easily to synthesize the fungal pathogens with this approach.
Okay. All right. Well, we've just about run out of time. I want to thank Pin Yang again for a really interesting talk and a great webinar. I want to thank all of the attendees for your attendance and your attention. And once again, thank you all and have a great day.
Yeah,
So,
thank you, Josh. Thank you very
okay,
much.
thank you.
Bye
All
bye.
right. Okay,
Yeah,
bye,
thank you.
everybody.